Hello, and welcome to CRPS Contender, my complex regional pain syndrome education channel. Today's video is pain and probiotics, and we will be taking another step down our journal journey, exploring a role for microbiota in CRPS. And before we launch into that paper itself, I want to talk a little bit about the human microbiome so that we have an understanding of what we're talking about in relation to this paper. The human gut microbiome forms a complex ecosystem of beneficial and pathogenic bacteria, which is trillions of microbes large and affects virtually all aspects of health, regulating anxiety, mood, cognition, pain, appetite, our immune system, our endocrine system, our social behavior. All of this happens via the gut-brain axis, which happens through our autonomic nervous system and our hypothalamus-pituitary axis. The beneficial and pathogenic bacteria have a balance that is impacted by internal and external factors. The gut microbiome produces short-chain fatty acids, and these have multiple effects. They're a source of energy, they're important in homeostasis, they regulate our immune response as well as our inflammation levels, they impact our appetite as well as our central and peripheral nervous systems. They are related to the integrity of the blood-brain barrier as well as our intestinal barrier. They interact with our hormone levels and electron transport chains, as well as our oxidative stress pathways. Point being, it does a lot of stuff, and the microbiome has a very active relationship with the host, which is you, affecting our gut-brain signaling, and is implicated in a bunch of different neurological disorders. There is a nutritional approach that is being promoted, particularly in this paper, but as well as just in general, to help prevent, ease, or delay these kinds of disorders, especially neurological disorders. So let's talk a little bit about short-chain fatty acids before we launch into the paper itself. Short-chain fatty acids are the primary metabolites of microbes. They're produced in the colon and play a key role in the neuroimmunoendocrine regulation, so nerves, immune system, and hormones, via the bidirectional vagus nerve signaling, as well as modulation of the immune system, the hypothalamus-pituitary axis, and tryptophan and tyrosine metabolism. Tryptophan ends up becoming serotonin, and tyrosine becomes your catecholamines, but specifically we're focusing on noradrenaline here. Neurological stressors can affect gut motility, the secretion of fluids in your gut, blood flow, your pain reception, and your immune system. This impacts your gut health, your intestinal barrier integrity, your mucus production, and your protection against inflammation. It also impacts your cerebral inflammation, your neuronal function, the microglia activation, which are the immune cells in your brain, and your blood-brain barrier integrity, which protects your brain from pathogens that are circulating in your blood. Short-chain fatty acids also modulate the levels of glutamate, glutamine, GABA, serotonin, catecholamines, appetite-regulating peptides, and intracellular potassium. There is an intestinal bacteria imbalance that is implicated in several disorders, especially neurological disorders. Okay, now that we have a little bit of background, let's launch into A Role for Microbiota in Complex Regional Pain Syndrome, which was written by Laura Crock and Crock Baldridge from Washington University in St. Louis, Missouri. It was published November 25 in 2020 in Neurobiology of Pain. Now this is just an excerpt from their abstract, not the whole thing. However, previous work has demonstrated that a subset of patients with new-onset CRPS, approximately 50%, improve if treated within 1 year, while the rest have minimal to no symptom improvement. Unfortunately, this leads to permanent disability and often requires invasive and costly treatments. Interestingly, there are case reports of remission or resolution of CRPS symptoms with the use of antibiotics known to affect the gut flora. We hypothesize that the variable clinical potential for recovery and response to therapy in CRPS may be secondary to or reflected in changes in the gut microbiota. We suggest that the microbiota may mediate or reflect clinical status via the metabolome, activation of the immune system, and/or microglial activation. We hypothesize that the gut microbiome is a potential mediator in development and persistence of CRPS symptoms, and propose that the clinical condition of CRPS could provide a unique opportunity to identify biomarkers of microbiota and potential therapies to prevent pain chronification. Now, what they're doing in this paper is they're collating a bunch of information related to the microbiota, not necessarily specifically about CRPS. So I have kind of pulled what I thought was the most important and the most relevant to CRPS specifically, but they actually reference way more than I talk about here. I just want to make that clear. So, the introduction and treatment of CRPS. CRPS is a devastating, persistent neuropathic pain condition occurring most often following stressful events and is often initially mistaken for an infection. Traditionally, CRPS is considered acute if the onset has been less than 12 months and chronic if the onset has been greater than 12 months. The development of CRPS is life-altering, and patients report decreased mobility, an inability to work, poor quality of life, depression, anxiety, and often long-term opioid usage. Sadly, conservative therapies and pain management fail 20 to 50% of patients with onset of less than 12 months, and they progress to the chronic condition of CRPS. Treatment efficacy decreases further as the disease becomes chronic. The prognosis. In the absence of significant co-founders such as concomitant psychological or neurocognitive disease, greater than 50% of patients with CRPS experience significant functional and symptomatic recovery. This recovery most commonly occurs early in the disease, and it is uncommon after a year or more of symptoms. While cytokine levels are higher in blister fluid of the affected limb with less than 12 months onset of CRPS when compared to the unaffected limb, these biomarkers have no predictive value for chronic CRPS, nor do we have biomarkers to predict which patients are at risk for developing or have potential to recover from CRPS. Identifying factors that contribute to CRPS recovery are critical to potentially preventing and/or curing this debilitating condition. The authors propose that the heterogeneity of CRPS symptoms and response to treatment may be related to microbiota differences. Let's talk about the microbiota and pain. Preclinical work in mouse models has shown that the presence of an intact gut microbiota is necessary for the development of normal visceral pain sensation. Decreased gut bacterial diversity and abnormal composition is associated with various painful conditions like irritable bowel syndrome, neuropathic pain, chronic pain, and inflammatory pain, as well as major depressive disorder. While no clinical studies have evaluated prebiotics or probiotics in the treatment of neuropathic pain, bifidobacteria and lactobacillus probiotics have been beneficial in treating IBS. And probiotics, as well as Planetarium 299V combined with an SSRI, have improved cognitive functions in major depressive disorder. There are case reports of chronic CRPS patients being placed on antibiotics known to alter the gut microbiome for an unrelated condition, whose CRPS then recovered after a treatment course. Discussing metabolites and pain. Gut bacteria are thought to be primary producers of short-chain fatty acids, which modulate inflammation, chemokine production, which is a subset of cytokines that interact specifically with white blood cells, regulation of tight intestinal barrier junctions, which is what keeps things inside your guts from going into your body, blood-brain barrier integrity, which protects your brain, and as well as having an important role in microglial homeostasis. Network modeling and pathway mapping can identify metabolite roles and aberrations. Important metabolic and taxonomic biomarkers for pain can lead to mechanistic insights and proof of concept with manipulation of these pathways. In a fibromyalgia cohort, serum metabolomics correlated with reduction in microbiome diversity. Understanding the gut composition of the microbiota in CRPS will clarify existing differences between acute CRPS versus chronic CRPS patients. Now let's discuss the microglia. One potential mechanism of gut microbiome influence on persistent pain is through the modulation of microglial reactivity. The microglia, which are non-neuronal brain cells that function as immune cells, make up about 10% of the total central nervous system cell population, and the microglia's functions include initiation of programmed neuronal cell death, clearing away of dead cells, the remodeling of synapses, brain injury repair, maintaining brain networks, and regulating brain development. Germ-free mice have developmentally immature microglia, suggesting that the presence of an intact gut microbiome is necessary for the functional maturation of microglia in the central nervous system. Manipulation of the gut microbiota in mice alters extinction learning through microglia-mediated synaptic pruning. Microglial activation has been implicated in the development of chronic pain and specifically in CRPS. Mechanisms by which microglia modulate the persistence of pain are unknown, and the neuromodulatory signals from the gut microbiota are a potential mediator. And now discussing inflammation. Through interactions with symbiotic bacteria, the immune system learns inappropriate and appropriate responses, as well as its ability to initiate local inflammation responses and mount antibody production. Microbiota plays broad roles in humoral immunity, which includes things like B cells and pro-inflammatory T cells, as well as regulatory T and B cells, and may contribute to autoimmune disorders via antibody production. Studies have implicated local immune responses or inappropriate post-traumatic inflammation in the development and persistence of CRPS. There is preclinical support for immunoglobulin G and M autoantibodies in CRPS. In one small cohort of CRPS patients, 55 to 90% of those patients had autoantibodies for immunoglobulin G. Although purely theoretical, it is possible that the gut microbiota functions to prime the immune system in CRPS, resulting in an exaggerated response to an injured limb. In conclusion, the authors of this paper provide a review of the latest advancement in CRPS research and propose a potential role for gut microbiota as a potential biomarker for diagnosis, treatment, and clinical course prediction. Current preclinical and clinical data support a highly testable role of the microbiota as a potential risk factor for development and persistence of CRPS. And again, I really cut down their paper because they had a bunch of other things in there and I wanted to keep this short. So that closes their section. I do want to make a few personal remarks. These are just coming from me. This is not related to these authors. Personally, before I was diagnosed, and after I was diagnosed, but especially before, and I did not know what was going on, I had severe intestinal pain and dysfunction. It was very unpleasant, and I spent a lot of time in the bathroom, and it was very messy. I did not have a good time, and it got to the point where it was actually worse than my nerve pain in my leg, and that actually was pushing me over the edge towards losing my grip on life. This is just coming from me. But if you are shedding your intestinal lining into the toilet when you're going to the bathroom, this is going to look like a tawny or a clear mucus. This is where your microbiome lives, and it shouldn't be shedding visibly. You should not be able to see it when you look down in the toilet. You cannot digest food without the beneficial bacteria in there to break it down. It will be very painful to excrete whatever you have eaten. So if you're experiencing this, you need to replenish that microbiome in there, or you're just going to keep going in the same pattern, especially if you're continually shedding. So you're going to want foods that have live probiotics in them. A lot of these foods are often fermented. Things like Greek yogurt, you're going to want to make sure it doesn't have added sugar in it, but like yogurt, kefir, tempeh, kimchi, kombucha, all these kinds of things have live probiotics in them. You can also take a supplement of a prebiotic or a probiotic. You're going to want something that's multi-strain, right? Because you're trying to increase the diversity of healthy, beneficial bacteria in there. You're going to want something with high CFU because, again, you're very depleted. You might do this in combination with an SSRI if you're really feeling very depressed or having, like a major depressive disorder as part of your CRPS development, or if you had it before. Personally, this really improved my quality of life. So I want to recommend it to you if you are also struggling with having a very bad time in the bathroom. So that's just my 2 cents in relation to this paper. Thank you very much for watching. If you'd like to check out any of the sources that I used for this paper, they're going to be linked in the description along with access to the slides. Please help me engage the algorithm if you like what I'm doing here so I can reach more people. If you'd like to contribute to my living expenses, my Patreon is CRPS Contender. And thank you to my patrons, Emily Malcontent and W4 Shep. I really appreciate your support. Alright, I think that's everything for me today. Thank you so much for watching. I hope you learned something today. And I hope to see you next time.